So this is the lecture video for experiment 5. There's a few key things that I want to touch on in this lecture video. It is also important to note that this reaction has already been started for you. So if you read and begin the pre-lab, you might be somewhat confused that it says that you were setting this experiment up in the previous week. We have set up all of these experiments for you such that they will incubate for approximately a week. So in your pre-lab write-up, you can make note that these were set up by the laboratory staff. Here is the overall reaction that's going to occur in this experiment. We are going to take ethyl acetoacetate, and then we are going to incubate it with baker's yeast under fermentation conditions, and we're going to make 3-hydroxy butanoic acid, their ethyl ester of 3-hydroxybutanoate. There's a couple key things to keep in mind here. The first thing that's important to keep in mind is that the reaction that's being catalyzed is a reduction, right? So we're reducing a ketone to an alcohol. Another thing that is a key thing to be aware of is that we are generating a chiral center during this reduction of a ketone to an alcohol, and as denoted in this scheme, this is going to be a chiral reduction. So most reactions that happen in biological systems are all going to be chiral reactions, that even if you start out with an achiral substrate, you're going to get a chiral product out. And that is absolutely the case in this system. There's a few things that are important to cover in terms of background information for understanding this reaction. I'm going to talk about it here. The key thing that's important to have a little bit of a handle on is the process of glycolysis. And so this is shown in your experimental description, and it is also covered in the lecture slides, which I'll switch over to in just a moment. So you are not responsible for understanding all the steps of glycolysis, but I'm just going to go through them very quickly just to give you a general sense of what's going on. And again, an important thing to keep in mind here is that we are doing this reaction under fermentation conditions, which it means anaerobic conditions, no oxygen. So these are the types of conditions where you generate, if you were doing fermentation for beverages, you would be generating ethanol. So the first step of converting glucose into molecules that can be taken forward in terms of energy metabolism is phosphorylation to give you glucose 6-phosphate. So you take glucose and then you phosphorylate this position here. So that does a few things. First of all, it traps the glucose in the cell, and it does a variety of things. Again, this is not really the forum for going through the details of glycolysis. The second step is you have an isomerization of this aldehyde to a ketone. The third step is that you have a second phosphorylation event, and that's going to drive this retroaldol cleavage to give you dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate will then be isomerized into dihydroxyacetone phosphate. And then this is really the key step that's so important for why under fermentation conditions that you will generate alcohol. And that is, in order to go through glycolysis, you have to take this NAD cofactor. and convert it to NADH. And this will be used later by the cell for energy metabolism. But this is really the key, that in order to proceed through glycolysis, you need NAD plus, and that gets converted to NADH. So then you go to 1,3 bisphosphoglycerate or diphosphoglycerate, and then again you go on down the pathway. So when you go to 3-phosphoglycerate, you actually generate ATP, and then you go to 3-phosphoglycerate, then to 2-phosphoglycerate. That gets converted into a molecule called phosphoenolpyruvate. That'll convert ADP to ATP. So because you're generating two molecules of dihydroxyacetone phosphate, when you go through here, you get two molecules of ATP out and two molecules of ATP out here when you get to pyruvate. The key here is that when you get to pyruvate under normal aerobic conditions, this can then go into something called the Krebs cycle, and then the Krebs cycle will generate all of the key materials that go onto the electron transport chain that go through normal aerobic respiration. But when you're under anaerobic conditions, in order to keep going through glycolysis, you have to be able to regenerate this NAD cofactor. And what you're going to find is that the key step for regenerating that NAD cofactor is you're going to convert acetaldehyde to ethanol. So this gets us now back into the lecture slides for experiment 5. I'm going to switch over into my tablet now, so hopefully there won't be any. Alright, so the exact reaction that we are going to be doing here is we are going to be utilizing the alcohol dehydrogenase that exists within yeast cells. So alcohol dehydrogenase is the main enzyme which is responsible for converting acetaldehyde to ethanol. There's two methods that you could potentially use. You could either do in vivo, which is the one that we're going to do, or in vitro. So in vitro, what you would be doing is you would be using purified enzymes, and you would try to mimic the conditions that exist inside of a yeast cell. That is not what we're going to do. We're actually going to do these reactions in vivo. And the way that this occurs is that the substrate that we're using, this ethyl acetoacetate, is permeable through the cell membrane. So ethyl acetoacetate can pass into the yeast cells and can be reduced inside of the yeast cells. So the actual method that we'll be using is in vivo. Alright, so what are enzymes? Again, without going into too much of the biochemistry of the subject, is that enzymes are typically large molecules. They're globular proteins. They can be as small as 5,000 molecular weight units or 5 kilodaltons, and they can get as large as several million molecular weight units. Enzymes are part of the superfamily of proteins, and consequently proteins are made up of polymers of amino acids. This polymer of amino acids will fold into a three-dimensional structure, and it is inside of this three-dimensional structure that catalysis will actually take place. And there's several other points here that you can read. I'm not going to. Alright, so the other sort of key topic that we want to talk about in this experiment is the concept of what do enzymes do? And without going into a lot of detail here, the real thing that is important to glean out of this is that enzymes are catalysts. Um, when you think of a normal reaction that is occurring with that is like a normal organic reaction that we typically execute in the organic laboratory, the the key limiting factor here is that when these molecules react, it is by pure coincidence that a nucleophile is colliding with an electrophile at the correct geometry and the correct energy. What an enzyme is going to do is it's going to take both the substrate, the electrophile, and the nucleophile, and it's going to place them perfectly such that they can react very efficiently. And effectively what's going to happen here sorry, I'm just getting used to using this. Pointer options. I want my pen. So effectively what's going to happen here is that if we think about this as a normal energy diagram for a reaction, so if this is your activation energy of the normal reaction, what an enzyme is going to do is it's going to lower that activation energy. It's not going to do anything. Oops. It's not going to do anything in terms of changing the overall energy of either the starting material or the products, but what it really is going to do is it's going to lower that activation energy for the reaction, and it's going to make it happen much faster. So as already stated, that we're going to be doing these reactions in vivo inside of yeast. Um, why is this? It's easy, it's straightforward. Yeast are very well studied. There's a variety of reasons for it. We've already gone through this idea of glycolysis, and it's important to, again, um, bring forth this idea that in order for the cell to go through glycolysis, you have to be able to regenerate this NAD cofactor. Okay, that's a real key point um, as to why it is that under anaerobic respiration, when you don't have oxygen present, you really need to have the ability to regenerate this cofactor. Okay. So now we're going forward. So again, here's the same reaction, just to reiterate. Once you make pyruvate through glycolysis, you're going to go through a decarboxylation reaction, and then you're going to get acetaldehyde out of this. And then acetaldehyde will react with alcohol dehydrogenase to regenerate your NAD cofactor and make ethanol. Until you are back into an aerobic system, this will be a metabolic endpoint. So this is the reason that when you do fermentation, pretty much all of your fermentable sugars will end up as ethanol. For this reason is that the cells still need to generate their energy currency, which is ATP, but they need to be able to regenerate this NAD cofactor. Normally, when you're under aerobic respiration, what happens is NADH goes to NAD when it's feeding the electron transport chain. But when you don't have oxygen present to get reduced to water, then this isn't possible. So under normal aerobic respiration, the key here is that you're going to go to acetyl-CoA, but again, under anaerobic conditions, we're going to go to ethanol, and we can only generate 2 molecules of ATP. So to compare what's going on in the cell with alcohol dehydrogenase to chemistry that you're familiar with from either 223 or 224, this reaction with alcohol dehydrogenase would be analogous to a sodium borohydride reduction of ethanol. So if you think about this, if NADH going to NAD is pretty much analogous to acetaldehyde going to ethanol using the reagent of sodium borohydride, then it's reasonable to consider that mechanistically it's probably going to be not that dissimilar. So thinking about the mechanism of how sodium borohydride can react and reduce aldehydes, we see how sodium borohydride can give up a hydrogen as a hydride, and then you get actual reduction of this molecule with the non-worked up final product, and then this just goes through the final steps of it. So in thinking about this from an organic synthesis point of view, your hydride can approach from either face of this carbonyl, and you can get either an enantiomer of the alcohol out. The appropriate terminology for this is called the C face and the re face. Again, I don't really want to go into the detail of this, but the C face, if it approaches from the C face, it'll give you one enantiomer. If it approaches from the re face, it'll give you a different enantiomer, i.e. the re face will give you the R, the C face will give you the S. So I don't know if that came through on the video, but it's Sunday night and my daughter wanted me to put her to sleep. So in thinking about how this reaction occurs with sodium borohydride, if we need an analogous type of reactant for ethyl acetoacetate, this is the key molecular component of the NADH and then NAD plus cofactor. And so if you compare these and just thinking about what is the difference in NADH versus NAD plus, hopefully you'll see that the key aspects of it are right here. And if we think about how this would potentially react, drawing our lone pair here, that's going to react there, and that's going to come up, and then that's going to come off as a hydride in a very analogous way to how sodium borohydride can lose a hydride. Okay. And then once that is lost as a hydride, then you go to the NAD plus cofactor. Okay. So this just kind of states what I've already said, is that in biological systems we don't have sodium borohydride, but the key reagent for doing these types of oxidations and reductions is NADH. Okay. So this just shows what I've talked about already, in that if you're starting here with this lone pair, and then this can come off as a hydride to give you a molecule that looks like this, and then how does that end up accepting hydrides when you go from alcohol to either ketone or aldehyde? Well, this drawn is this resonance structure to explain that. So this just again goes through mechanistically how this is happening, where you have your ketone or aldehyde that's going to react with NADH. Your electrons are going to come up, your hydride is going to come over here, and then you're going to have your reduced product here. To show the reverse reaction of how you would actually generate NADH through the oxidation of a molecule, that you would have your NAD plus cofactor, and then you'd have a base that would come off and then remove this hydrogen ion. Those electrons would come down here to form this carbonyl, and then this hydride would kick off. So that this shouldn't look like a good reaction to you. This should look like a if you were to draw this reaction on an organic chemistry exam, it would not be considered well by the instructor or grader. And that is because the only reason this can happen, and we'll get into this in just a couple slides, is that these reagents, this reaction is positioned perfectly inside of the enzyme active site. It's also a nice piece of trivia to understand in this case that if you do ingest alcohol, so this is the example of ethanol, and the enzyme that would catalyze the metabolism of ethanol in your body, the first step would be via alcohol dehydrogenase, the enzyme that we're dealing with in this experiment, where you would start out with ethanol, you would go to acetaldehyde, which again, a nice piece of trivia for you is that if you are ever around individuals who have been drinking significantly, they always have a very distinct smell to their breath, and this is the molecule that is responsible for that smell. This will then go on further if under aerobic conditions to generate acetic acid, which will get converted to acetyl-CoA eventually, and then acetyl-CoA will go through the Krebs cycle and be pushed through energy metabolism. So don't let anybody tell you that drinking ethanol is all bad. Okay, so unlike sodium borohydride, and we've already alluded to this reaction is stereospecific, i.e. that there is an enzymatic active site that only holds the substrate one way and that the cofactor is positioned such that it can only react in one way. This is what this is sort of a diagrammatic representation of the active site, is that you're going to have a histidine that's going to hold a zinc ion, and that's going to position the carbonyl such that it can react perfectly with the NADH cofactor. The only difference here is that we are going to utilize ethyl acetoacetate instead of acetaldehyde, and so this is going to bind in the active site very similarly to the way that acetaldehyde will, but it's just going to have this extra peripheral cabbage on it. Okay, so I'm going to go through these last slides pretty quickly. This is a three-dimensional diagram of what the enzyme active site is going to look like and how the ethyl acetoacetate is going to fit in there. I'm just going to kind of skip through here. You can walk through these slides if you want. But there's going to be a couple things that are going to be important for us to do. We're going to use infrared spectroscopy to determine if the reduction occurred. So the key thing that we'll be looking for there is we're going to be looking for the disappearance of a ketone and the appearance of an alcohol. So hopefully you will have already looked into this and known what to be looking for in terms of what signal you'll lose with your ketone and what signal you'll see with your alcohol. The other thing that's important to bring forth is this idea of optical purity. So because we are generating a new chiral center, that once we isolate this product out, it will have a the ability to rotate plane polarized light. And the key calculation that you're going to be required to do is to be able to identify what the optical purity of your product is. So it's your observed specific rotation divided by the known specific rotation of the pure enantiomer. And all of this information is in your lab description. Okay. And then in addition, you're going to have to know how optical purity is related to your enantiomeric excess and what the enantiomeric excess is. Okay. All right. That is all for this lecture.